Dear students, the topic is bacterial growth curve.
Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment.
When a broth culture is inoculated with a small bacterial inoculum, the population size of the bacteria increases, showing a classical pattern. When plotted on a graph, a distinct curve is obtained, referred to as bacterial growth curve. Bacterial growth curve can be obtained by growing a pure culture of bacteria in a liquid medium at a constant temperature. Samples of the culture are collected at fixed intervals, example every 30 minutes, and number of viable organisms in each sample is determined. The data are then plotted on logarithmic graph paper. The logarithm of the number of bacteria per milliliter of medium plotted against time. In a bacterial growth curve, there are four phases: lag phase, log phase, stationary phase, and a decline phase or death phase. We can see one by one stages of bacterial growth curve in detail. This figure shows the bacterial growth curve, number of cells and time. See, the four phases are shown here: lag, log, stationary and decline phase. The first phase is lag phase. After a liquid culture broth is inoculated, the multiplication of bacteria does not start immediately. It takes some time to multiply. The time between inoculation and beginning of multiplication is known as lag phase. In this phase, the inoculated bacteria become acclimatized to the environment, switch on various enzymes, and adjust to the environmental temperature and atmospheric conditions. During this phase, there is an increase in size of bacteria but no appreciable increase in number of bacterial cells. The cells are active metabolically. The duration of the lag phase varies with the bacterial strains, nature of culture medium, incubation temperature, etc. It may vary from 1 hour to several days. The second phase is log phase. This phase is characterized by rapid exponential cell growth, that is 1 to 2, 2 to 4, 4 to 8 and so on. The bacterial population doubles during every generation. They multiply at their maximum rate. The bacterial cells are small and uniformly stained. The microbes are sensitive to adverse conditions such as antibiotics and other antimicrobial agents. Growth rate is greatest during the log phase. The log phase is always brief unless the rapidly dividing culture is maintained by constant addition of nutrients and frequent removal of waste products. When plotted on logarithmic graph paper, the log phase appears as a steeply sloped straight line. The third phase is stationary phase. After a log phase, the bacterial growth almost stops completely due to lack of essential nutrients, lack of water, oxygen, change in pH of the medium, etc., and accumulation of their own toxic metabolic waste. It is during this phase that the culture is at its greatest population density. However, death rate of bacteria exceeds the rate of replication of bacteria. Endospores start forming during this stage. Bacteria become gram variable and show irregular staining. Many bacteria start producing exotoxins. Then the fourth or the last phase is decline phase or death phase. During this phase, the bacterial population declines due to death of cells. The decline phase starts due to accumulation of toxic products and autolytic enzymes and exhaustion of nutrients. Involution forms are common in this stage. Some cells assume various shapes, becoming long, filamentous rods or branching or globular forms that are difficult to identify. Some develop without a cell wall and are referred to as protoplast, spheroplast or L-phase variants or L forms. When these involuted forms are inoculated into a fresh nutrient medium, they usually revert to the original shape of the healthy bacteria. Next, we can see the significance of bacterial growth curve. The study of bacterial growth curve is important, aiming to utilize or inoculate known numbers of bacterial isolate, for example, to enhance plant growth, increase biodegradation of toxic organics, or produce antibiotics, other natural products at an industrial scale. Knowledge of bacterial growth kinetics and bacterial numbers in a culture medium is important from both research and commercial point of view. Growth kinetics is also useful for assessing whether particular strain of bacteria are adapted to metabolize certain substrates such as industrial waste or oil pollution. Bacteria are genetically engineered to clean up oil spills, for example, can be grown in the presence of complex hydrocarbon to ensure that their growth would not be repressed by the toxic effect of oil. Similarly, the slope and shape growth curves produced from bacteria grown with mixtures of industrial waste products can inform scientists whether the bacteria can metabolize the particular substance and how many potential energy sources for bacteria can be found in the waste mixture. Dear students, thank you for watching. See you in next class with another topic. Don't forget to leave a like and please subscribe my channel.